Welcome to the Francis Crick Institute, and hello to people joining us online as well. My name is Claudia Hammond, and I'm the presenter of Health Check and the Evidence on the BBC World Service, which are both global health programmes, and also All in the Mind on BBC Radio 4, which is a programme about neuroscience and psychology and mental health. So it is lovely to be here again for World Immunology Day. Each year we talk about a different topic here, and we have amazing panels, and it's set to be another fascinating evening, because today we're looking at how the immune system can affect the brain and the nervous system. And each year we gather here, as I said, to celebrate, in a way, a different topic. And today's event is a joint venture between the Crick Nature Portfolio and the British Society for Immunology. I've been asked to fill you in on what they all do. The Francis Crick Institute, where we are now, you may well know, is a biomedical discovery institute which researches all the biology underlying human health. And so above our heads and beneath our feet, scientists are working and trying to work out the basic biology and then translate those discoveries into new treatments, new ways of preventing and diagnosing the diseases that may or may not affect us all. But there's lots for the public to get involved with too. You might have seen the exhibition when you were waiting outside, and there's a lot going on in person and in schools as well. Nature is one of the leading science journals. You often hear about it on the news because it's often where really groundbreaking papers on brand new discoveries are published, and there's lots of other journals in their portfolio too, and some of their editors are here tonight. And the British Society for Immunology represents the scientists and clinicians who are studying the immune system in health and disease. And of course, this is all about people, all about our immune systems and all about people. So key to all of this is public understanding and engagement, and that brings us to where we are tonight. So what's going to happen is there'll be a short talk, first of all, from one of our scientists who'll be on our panel with an introduction to some of the basics of what we're talking about. Then we'll have a discussion, and there will be two separate opportunities for you to ask questions and for people at home to put their questions into the Q&A and to ask those as well. We really have some of the best experts there are on this topic who've made some really groundbreaking discoveries of their own. So if you have a question, this is the place to ask it. The rule is no question is too stupid. There is no such thing. You're allowed to ask anything. So first of all, we're going to hear from Dr. Lorena Aranthibia, who works here at the Crick and at University College London as well. She'll explain a bit more later on about exactly what she does there. But she's going to give us a short primer on how the immune system is linked to the brain and the nervous system and which diseases can result. Thank you, Lorena.
When we think of our brain, we think of our command center. And when we think of the immune system, we think of our defense force. One thinks and the other one protects. But in recent years, we've learned that these two systems are more than just neighbors in our body. They're partners, and they're in constant communication, influencing each other's decisions. And as we'll find out today, sometimes they can misunderstand each other. So today we're going to explore what happens when the immune system meets the brain. So let's start with the immune system. At its core, it's a sensing and responding network, a kind of internal surveillance force. It's an army of superheroes that's constantly scanning for signs of trouble, infections, or anything that doesn't belong. And it does this using a large number of different cell types. So let's meet some of these superheroes. We have neutrophils, dendritic cells, and macrophages. They're all part of what we call the innate response. They're the first-line defenders. They act quickly to kill or eat any pathogen that may be found in the body. And importantly, they raise the alarm as well, calling in reinforcements. And that reinforcement comes in the form of B cells and T cells. These are part of what we know as the adaptive response. This is a slower response, but it's smarter. So B cells can produce antibodies that help tag pathogens to make it easier for macrophages to eat them, and T cells kill infected cells. And what's really important about these types of cells is that they have a memory. They can remember and offer long-term protection. And this is the basis for why we don't get an infection twice and why vaccines work. these cells don't work alone. They also rely on soluble factors. And so we have complement. This is a large set of proteins that work together in a cascade and help tag intruders and boost inflammation. And we also have cytokines and chemokines, which are messengers between these cells and help guide the cells towards the site of infection, kind of like a homing beacon. And together, these two systems help direct traffic, amplify or resolve inflammation, and maintain balance. Now let's talk about the nervous system. Interestingly, I can use the same words to describe the nervous system as I use to describe the immune system. It can sense and respond. It processes information from outside and inside the body to guide everything that we do, our movements, our decisions, our emotions, our feelings. And we often divide it into the central nervous system, which is the brain and the spinal cord, and the peripheral nervous system, which is the vast network of nerves that extend from the brain and spinal cord all the way to the tips of your fingertips and toes. But remember, this is one system. It's seamlessly connected and constantly talking. And today we're going to be zooming in on the brain. This is the most complex and beautiful organ inside your body, and it's our control center. It is very powerful, but it's also very delicate. So delicate, in fact, that for many years we thought that it was what we call immune privileged. This means that none of the cells that I've just introduced to you as being part of the immune system were ever found inside the healthy brain. It seemed like the brain was locked in a fortress, so protected that it was shielded from pathogens as well as our own immune system. We're now learning that this isn't quite true. The brain isn't immune silent. It's still immune privileged, just not silent. It's immune special. So how does the brain keep itself protected but stay in touch with the immune system? Well, it doesn't rely on just a single line of defense. It relies on a network of borders. So we have some here. We have the choroid plexus, which is a structure found inside the brain ventricles, which are gaps inside the brain, important for producing a liquid called cerebrospine fluid. And in this fluid, we find that there's a high level of immune cells and immune molecules. We also have the meninges. These are layers of tissue that wrap around the brain. And again, we have a high concentration of immune cells there. We've even recently found that the skull, which we know protects the brain by allowing this huge density of bone to stop the brain from getting injured, also has a bone marrow, and it can produce immune cells, such as B cells, and has little channels that communicate directly with the meninges to deliver B cells whenever they're needed by the brain. And perivascular spaces are fluid-filled channels around the vasculature of the brain, and they normally act to help drain waste from the brain, but also act as communication highways between the brain and the immune system. And together, these regions act like border patrol systems, filtering, monitoring, and sometimes responding to the immune system. And the most famous of these borders is the blood-brain barrier. So behind me, you can see the vessels of the brain. They're labeled in green. These are vessels, very thin capillaries that extend throughout the brain. And we have astrocytes labeled in red. And you might be able to see that they have very fine processes that wrap around the vessels. And if we look at a cross-section of it, the blood-brain barrier is a tightly sealed lining of the vessel being filtered by astrocytes, pericytes, and endothelial cells. And together, these cells form like a selective membrane. The selective filter allows the entry of oxygen and nutrients into the brain, but also stop any large things like pathogens or any toxins from going inside the brain. And what we now know is that this blood-brain barrier can change over time in development, in aging, and even in disease. And it can adjust to the brain's need and even its stress signals. So we've crossed the border, so now let's meet the cells inside the brain. We have neurons. When anyone thinks of the brain, they immediately think of neurons. These are cells that communicate by sending electrical messages between each other. But they don't work alone. We also have oligodendrocytes. They make a fatty membrane that wrap around the processes of neurons, insulating the brain from this electrical signaling. But they also help speed up the messages between neurons. Astrocytes I've already briefly introduced as part of the blood-brain barrier, but originally they were thought to be scaffolding in the brain, sort of shaping the structure of the brain. And we know that they do much more. They support neurons, and even though they're not immune cells, they can respond to injury and call for reinforcements. Pericytes, they're strictly not brain cells. They can be found elsewhere in the body, but they do carry out an important role in the brain. They maintain and guard that blood-brain barrier that I talked about, and they also regulate blood flow. And our star of the night is microglia. These are the brain's very own resident immune cells. They're macrophages, and they work as immune sentinels, constantly checking out what's going on in their surroundings. And you can see some of these in these videos here. You can see that microglia are these beautiful cells. They have long processes that are branched, and they're constantly moving those branches, checking out what's going on in the brain. Each cell has its own surveillance zone, and they can also react very quickly. So when there's an injury, as it will be shown in the video here, they quickly move those branches to the injury site and then start moving the whole cell bodies and asking for help, pulling in other microglia to the site. So microglia also have other jobs in the brain. They're the brain macrophages, but they wear many hats. So they're the patrollers that I just said. They're scanning for any injury or disease. They are also the cleanup crew, so they can also clean up any dead cells that may be found in the brain or any debris or toxins. They're also therapists. They send out nice immune factors to the brain, telling the brain that everything's going to be okay. And they're also architects. They play an important role early on in development, guiding the neurons to allow those connections to form between the cells and developing synapses, which are the contact points between neurons, and also getting rid of any excess synapses that are no longer needed as we develop. And this really changes that old idea that the immune system is there to protect us. In the brain, it also shapes, maintains, and fine-tunes neural function. Like any powerful system, microglia can also cause harm. So when they're overactivated by stress, aging, or disease, they become pro-inflammatory, which means that they start releasing those damaging cytokines, which can hurt nearby cells. They also start getting rid of healthy synapses and breaking down myelin. And when they do this, they change their shape. They sort of retract all the processes and become these blobs, which make that they're less effective as the guardians. And this brings us to neuroinflammation. So what do I mean by that exactly? Neuroinflammation is an immune response anywhere in the brain or the spinal cord. It can be helpful in the short term, so it can stimulate brain repair when there's been a damage or an infection. But if inflammation becomes chronic or misdirected, it can cause lasting harm by killing neurons, damaging synapses, or disrupting communication between different brain areas. And we now know that neuroinflammation plays a key role in a wide range of diseases. So it plays a role in diseases including autoimmune diseases like multiple sclerosis, in psychiatric disorders like depression or schizophrenia, and in neurodegenerative diseases like Alzheimer's and other dementias. And they don't just play a role in this. We now know from genetic studies that many of the genes linked to the risk of these diseases include genes involved in microglia function, in complement, and cytokine signaling. And this tells us something very important. The immune system in the brain may not just be responding to disease, it could be part of what's causing it in the first place. So let's have a look at Alzheimer's disease as an example. In Alzheimer's, we have a buildup of a toxic protein called amyloid beta, and that leads to the formation of amyloid plaques, and those are shown there in pink. Now, for a long time, it's a neurodegenerative disorder, and we thought that we should focus on neurons and amyloid plaques as the problem. But microglia carry much of the genetic risk linked to Alzheimer's disease. And what we're finding is that they walk a fine line between being helpful and causing harm. So microglia react to amyloid plaques, and they help clear out that toxin as it's building up, but they also contribute to the formation of the plaques. when they're like overactivated by this plaque pathology, they start releasing damage in cytokines and chemokines, which damage nearby neurons or astrocytes, like the cell there shown in blue. And together with complement, they start a pathway that can tag synapses and lead to excessive synaptic loss, which we think may contribute to cognitive decline. So microglia aren't just bystanders in Alzheimer's disease. They may shape the disease itself, whether it's for the better or the worse, and there's much still to learn here. So hopefully, I've shown you today that the brain and the immune system aren't working in isolation. They're in constant conversation. And that back and forth shapes how we grow, how we age, and how we respond to disease. Things like stress, sleep, exercise, and even the gut microbiome can shape brain responses through the immune system in ways that we're only just beginning to understand. So this is a really interesting field of investigation, and it's really exploding with new ideas. And this, there's still a long way to go, but the more we learn about this, the more we can treat things. So we're now starting to develop things like immunotherapies for treating brain disorders, which we previously only thought were a problem with the neurons. So all this just brings us hope for the future, and I now look very much forward to our discussion. Thank you. Thank you.
Well, thank you so much, Lorena. That was absolutely fantastic, and you packed lots into that. So there's lots more we can unpick too as we go through. But I'd like to welcome my panel now and to give them the chance to introduce themselves. So, Lorena, first of all, can you explain what is it you do in your day job?
Hi, so yes, I'm Lorena. I work here at the Francis Crick, which is a lovely place to work. I am also part of the UK Dementia Research Institute and also a researcher at University College London. I'm a neuroscientist by training and only really moved into the field of neuroimmunology about 8 years ago when I started working with microglia. I now have a strong interest in Alzheimer's disease and the role that microglia play in developing the disease and the disease progression. And in particular, we're looking at the early stages of the disease and how microglia communicate with the other brain cells for this development.
Fantastic. And we also have Dr. Soyeon Hong.
Hi.
Can you tell us about what you do?
Yes, I can. Is my mic on? Yeah. So hi, I'm Soyeon Hong. I have my lab at UCL and UK Dementia Research Institute. And my lab studies microglia, which are the brain's major immune cells that Lorena talked about. Specifically, what I study is how my group and I, what we study is how microglia interact with the neurons, the critical cells that by which we think and we talk and we, so what we're studying is how the immune system, including microglia, impact neuronal function and neuronal health.
Thank you very much for that. And we also have with us Professor Paul Morgan. Paul, can you tell us about your job?
Yes, I'm Paul Morgan. I'm the director of the UK Dementia Research Institute at Cardiff. I should say that three of the panel, us three, are all from the UK Dementia Research Institute, which is spread out across UK universities and set up about 8 years ago now, and really has become a focus for dementia research in the UK. So an advert there for the DRI. So what do? I'm an immunologist by training, and I worked for all of my long career in inflammation, and particularly on what Lorena mentioned, that bit of inflammation called complement, the complement system. And I got pulled into dementia, really, having worked in many other different diseases and roles of inflammation in them, with the foundation of the Dementia Research Institute, where evidence was starting to accumulate, again, as Loretta mentioned, that neurodegenerative diseases were more than just brain rot, they were actually inflammatory diseases of the brain. So suddenly this became an area of real interest to me and others interested in inflammation.
Thank you very much. And lastly, we have Professor Jessica Teeling. Jessica, can you tell us about what you do?
Yes, I can certainly do that. Hello, my name is Jessica Teeling. I work at the University of Southampton. I'm an immunologist by training, And about 20 years ago, I had the opportunity to use some of my knowledge of the immune system on the brain. So we're particularly interested, my team and I, on how the peripheral immune system communicates to the brain. So what happens when you might have a bacterial infection or a viral infection. And so what we're looking at is how it might contribute to the onset and the progression of neurodegenerative diseases, and particularly Alzheimer's and Parkinson's disease. But I also have an interest in immunotherapies, which was introduced by Lorena. And so by using monoclonal antibodies, we can treat cancer. And I've done that in my past career. And what we're now interested is how to use those monoclonal antibodies for the treatment of diseases such as Alzheimer's disease.
Thank you. So as I said, we have this amazing panel. And Lorena, listening to your talk there, one of the things you can't help but do is just marvel at what is going on in the brain. And this is one tiny aspect of it that you're talking to us about. All these different types of cells with such specialist jobs. It is just sort of, I don't know, blow your brain looking at that. It's just marvellous, isn't it?
It is confusing, and it is something that has been developing really quickly over the last couple of decades. So it was genetic studies that started pointing us towards looking at the immune system in the brain. Microglia they were discovered just over 100 years ago, so they're not easy to look at, but they were obviously the first thing that we wanted to look at. But in the brain, like in Alzheimer's disease, we have a lot of different cells that are responding. And so microglia also starts responding in ways that they start releasing these cytokines, things like IL-6, IL-1 beta, TNF. These are pro-inflammatory cytokines, and they influence everything. So they're not just there to call for help from other cells, but they're also affecting how astrocytes behave or neurons behave. And so it's just such a complicated system to look at, basically.
Yeah. It's spectacular, though. So, Paul, I want to start with you. And as Lorena was saying to us, we used to think that the immune system and the brain were completely separate. And I liked this idea that the brain is locked in a fortress. But actually, we know that the blood-brain barrier can let some things through. Is that a good thing and a bad thing? I mean, would it be better if it just lets less through? How does this all work?
Yeah, when I was a medical student, the idea was that the brain was encased in this completely impregnable barrier, the blood-brain barrier. So cells didn't get in, most proteins didn't get in, and when they did get in, it was a bad thing. And that really was a marker of pathology, that disease. You'd see it in diseases like multiple sclerosis, for instance, where proteins that should be outside of the brain were getting into the brain. What we now realize is, first of all, that barrier normally is quite selective and will let some things through quite efficiently by having transport systems for key molecules that need to get into the brain. But not just molecules, but cells. A lot of cells do traffic in and out of the brain. We've learned this, I guess, really quite recently. So we used to think that there were no T cells, for example, in the brain. We now know that's not the case. We now know that T cells do enter the brain from the periphery, even probably in health and certainly in disease. So it's much more linked to the periphery than we used to think it was, even in health. And once disease kicks in, we see a lot more immune activity in the brain than we might have expected even a decade ago.
And so, Jessica, could this go some way towards explaining why it is that, I don't know, when your immune system is fighting infection, that you might feel depressed or really tired or sick even, nauseous?
Yes, there's good reason to believe that you can explain that by the activity of the immune system in the body. And so when your body fights an infection, a number of physiological and metabolic and behavior changes occur. And we've all been ill, and we've all experienced, the immune to brain communication that happens, and it's an adaptive and needed response. So one of the very well-known responses is the production of fever, where you have an elevated body temperature that reduces the growth of bacteria, for example. But there's also brain areas that control your sleep, that control your energy levels, and that control your mood. And so when all these systems are activated to fight the infection, your brain is contributing to that by reducing your activity and improving your sleep and inducing some transient memory changes. It's adaptive, it's good, it's needed. The problem is when your brain has already got an increased level of inflammation, then the problem might occur, and that's what we see as well.
Yeah, so it's getting this balance right between the right amount of activity but not too much activity that's going on. Soyan, I wanted to ask you about these microglia, which I like to tell you called them the stars of tonight. So can you explain a bit more about what they do and how it is that they might shape, say, something like Alzheimer's disease and the type of it?
Yeah, so I remember about 20 years ago, my first meeting, scientific meeting I went to, seeing this video, which one of the videos that Lorena showed, that completely revolutionized our thought about these immune cells as microglia in the brain. So what scientists did about 20 years ago is to label microglia in living mice green. So they tagged it so we can visualize it using a camera through the skull of the mice. And what we observed was that microglia were remarkably active in a normal, healthy brain. Before, people thought that the immune system of the brain, which are microglial cells, they're probably the primary immune cells of the brain that reside in the brain. So microglia come in very early on when, during fetal stages, during embryonic stages, microglia come into the brain and they stay there. And people thought, traditionally scientists thought, that microglia don't do very much until there's disease or there's immune activation and so forth. But what that video, by visualizing microglia in the healthy brain, showed us is that microglia are remarkably active, right? So I think of microglia, going back to your question, as the first responders, right? The policemen, the firemen I guess police is the best way because they're continuously surveying, right? To make sure that the neighborhood is safe, right? The neighborhood watchers. So that's microglia's role in a normal, healthy brain. They're constantly moving around, trying to see whether there are any signs of injury or any invaders and so forth. And in the case when there is an invader or injury, they're the first to respond. So they play important roles in health as well as during disease, right? So what we're thinking in an age-related disease such as Alzheimer's disease, where aging is really important, right? So when our brain ages, when our body ages, what we're observing is that microglia do become old and tired. And so unfortunately, what we do see is that their response system is not as robust. And what we think is that this decline in the normal protective functions of microglia contribute to Alzheimer's disease, for example.
And it's amazing how new these discoveries are.
Yes.
That this is all so recent.
Yes, yeah, it's amazing time to be in brain science as well as immunology, right? Brain was particularly hard to study because, it's hard, we can't biopsy the brain easily, right? But now, thanks to technology, advance in technology, we're beginning to understand that the brain is, as Lorena described, quite multicellular and quite important throughout development and lifespan and aging as well.
And Paul, we heard mention there in the talk as well of the complement system. I know this is very much an area of yours, as you mentioned. Can you explain a bit more about how that works, what exactly complement is?
Yeah, so we've talked a lot about first defenders, and complement is the first defender par excellence. It's a system of proteins in our blood which are there to defend against bacterial infection and also to deal with garbage, to remove garbage. So people who are deficient in complement have bacterial infections and get them very severely and can die of those bacterial infections very early in their lives. And also, if they don't die of those, they get these immune complex diseases where the garbage accumulates in the tissues, and diseases like lupus you may have heard of. So that's what complement does. It does all that maintenance role, which without having that working, you get all these problems. So in the brain, what it does is the same sorts of things. It's dealing with the risk of pathogens, which shouldn't get there, but if they do, complement's there to deal with it, and also deals with the garbage which accumulates in the brain, probably more than anywhere else. And dealing with garbage like amyloid in the brain is really, really critical, and complement plays a role. And that's all the good things, but the problem with complement is it drives inflammation. And inflammation is great when it is controlled, but when it is in excess, then it causes damage. And so what we see in disease, not just in the brain, but peripherally as well, is inflammation driven by complement dysregulation, too much activation, can cause damage to the tissues, damage to the cells, and disease. So what we are trying to do in the brain is to inhibit the complement system, specifically to reduce that inflammation and therefore improve the outcome in people with dementias.
And why has the brain got so much garbage? You said it's got more garbage than anywhere else. Why is
Because
that?
it is quite a closed box. I think that's the real reason. So in another tissue, like, I don't know, the liver or muscle even, where garbage accumulates, macrophages get in there, they take out the garbage, and they're in the circulation. They can dispose of it very easily. They take it to the spleen or the liver, where there are very good systems for taking that and disposing of it. The brain doesn't really plug into those systems anything like as well. And so you need that integral system, which is really complement plus microglia to deal with the garbage locally.
So one way, it's not good that the brain is locked in this fortress, kind of.
With everything's a double-edged sword.
Yeah.
The barrier is critical to the function of the brain, but it also then, that comes at a cost. And some of that cost is not having access to the homeostatic roles of the peripheral immune system.
So Soyan, can the immune system even affect the brain while it's still developing? I don't know if it's a fetus or as a toddler child. Can the immune system make a difference then?
Yes, definitely. So I think I want to tie in with the complement system that Paul just mentioned, because so what was amazing is people thought complement is a defense system, which is true, but in the brain, our brain uses this complement tagging system as a way to clear up the neurons that we don't need anymore. So when our brain develops during embryonic stages and when you're a toddler, there's a scientific phenotype called brain wiring, right? And that's crucial for our brains to develop normally. So when you're first born, our brains usually have too many synapses. The synapses, as Lorena mentioned, are these important connection points between neurons. So initially, a baby's brain has a lot of connections that needs to be pruned away. So very simply put, our brain does gardening, right? And if you are a gardener who gardened, who took care of roses before, what is critical is to prune away the leaves that are not bearing fruit, and pruning is really important for correct, right, roses to bloom. And that's exactly what our brain does. And the real surprise came, again, not too long ago, less than 20 years ago, is that the brain actually uses complement as one of the molecules to regulate what to eat and what not to eat. And who eats it is microglia. So it's very fascinating.
Yeah, so that whole thing gets linked up at whatever age you are. Yes. And Jessica, we hear a lot these days also about the gut-brain axis, and I know this is something you very much work on. Can you explain exactly what that is, and does that mean that the state of our gut is actually affecting our brain?
The gut, brain, and immune axis, because they all three are very much related, is an absolutely fascinating connection to the brain. And again, it's only recently that we sort of realized that what happens in your gut has a strong connection with the brain, and your gut feeling is actually a real thing. Your gut can control how you feel. So the bacteria in your gut control many different physiological aspects. So they control how the gut barrier functions, so make sure it's not leaky and nothing comes out and into your bodies. It also regulates your energy levels. It can provide vitamins, and it controls your immune system. And so changes in the gut microbiota and the bacteria that live in there can really influence your physiology. And we now know that really influences how your brain functions.
And so do we know that the microbiome is changing your immune system?
It definitely does. Yeah, there's absolutely a critical link between the bacteria that live there and control physiological responses. And the immune system is very important. But there's more than that. Bacteria make neurotransmitters. Those are the chemicals that go from one neuron to the other. That makes them tick. Immune cells make neurotransmitters, and actually serotonin, which is the happy hormone, is produced more in the gut than in the brain, and is produced by bacteria and immune cells. And so these connections are super strong and super important in how our brain functions. And if you have a change or a dysregulation of your bacteria, a dysbiosis as we call it, it changes how you feel. It's connected to anxiety, depression, and now also dementia.
And Lorena, what about as we age, how does communication between the immune system and the rest of the nervous system change as we age?
So as we age, both the immune system and the brain undergo functional decline. And it's a process that, a condition that we know as inflammaging. And what happens actually is that you have this chronic sort of inflammation that builds up over age. And it has nothing to do with infection. It's just the immune system itself is changing and becoming more and more inflammatory. And this is critical with aging because it just means that everything's poised more towards that neuroinflammation, which means that we're more poised to be susceptible to immune diseases in the brain, such as the dementias and autoimmune disease. And there's also like that blood-brain barrier that I talked about, that changes with aging as well, and it becomes leaky, let's say. So it doesn't become as good as a selective filter anymore. And as Soyeon already mentioned, the microglia, they become more sluggish, they're not so good at cleaning up, and again, that affects our brain in aging. So actually, aging plays a key role in this communication between the different.
So it all gets worse, basically, as well.
Sadly, it does.
Telling us. Yes, this is not good, but yes. Well, we have some time for questions now. As I said, we're going to take questions twice. In the second half, we're going to talk much more about specific diseases and about what we might be able to learn to tackle those diseases from some of these findings that have been there. But is there anything people would like to ask on what they've already been hearing so far? If you would like to, then put your hand up. There is somebody up the back, and we have people with roving mics. Please wait for the mic to get to you. Yeah, there is somebody up there.
Hi, I'm a parent with 2 boys that have pandas. I'm not sure if hopefully you're familiar. No, so it's immune-mediated neuroinflammation. So as a result of a strep infection 4 years ago. So I'm interested to know whether your work whether you've come across this and how to turn it off, which I guess is your whole work, is try to turn off neuroinflammation. But once the trigger, i.e. the strep infection, has gone, whether your work with Alzheimer's or has come across this.
Is this a condition any of you have come across in particular? Yes, Jessica?
Yeah, I have come across, I don't work on it, but I have come across it. And it's an immune response or an unwanted immune response against neuronal elements in the body. And so, as I understand, there may be an antibody response against neuronal elements that shouldn't be there, and that you can see it as an autoimmune reaction. And so the Streptococcus bacteria is driving autoimmunity. So in general, our bodies avoid autoimmunity by training our immune system when it develops. Unfortunately, it doesn't get trained to antigens, that part of the nervous system, because of the immune privileged element. And if you get an infection, such as strep infection, you might get an increase in antigens that shouldn't be there. And if these are neuronal related, they can cause these diseases. And many bacteria and viruses, they have similar molecular structures to the nervous system. And if you do get an autoreactive reaction as an infection, it often influences, some neuronal element. How do you switch it off? Yeah.
Precisely.
It's not easy to switch it off, but there may be some anti-inflammatory therapies that can be used. For example, the use of intravenous immunoglobulins, which basically filters out the autoreactive antibodies as a normal reaction that you get rid of. So increasing high levels of antibodies that are not autoreactive will then flush out the autoreactive antibodies, but it doesn't really switch off the disease. It's more of a symptomatic treatment.
Thank you for that. Thank you for your question. Anybody else have a question at this point? Do wave. You were pointing to something. Oh yes, up there. Yes, thank you. The lights are bright, but do wait till the mic comes to you, please, so that everyone can hear.
Hiya. So you mentioned about how the microbiome can kind of affect the brain and how we feel and things. Is that why certain changes to your diet are supposedly linked to like not preventing dementia and things like that, but certain foods is meant to like help with your brain and things like that. Is it to do with like how they affect your microbiome?
Paul, yeah.
I think the answer is partially yes, and I think that we hear all these adverts for yogurts which are meant to give you this fantastic boost to your microbiome, and there's there's a little bit of evidence, but not very much, that that might have some significance. But in general, a healthy microbiome is a very good thing, and how you get a healthy microbiome is challenging. A healthy diet, on the other hand, is certainly protective against dementia, and good vascular health. And of course, if you eat a very fatty diet, full of junk foods, etc., then your vascular health isn't going to be good, and that's certainly going to impact on the brain as well, because all the dementias we see really are mixed dementias. They're not just purely Alzheimer's. They will have a vascular component, and that makes it a lot worse. So if we can maintain good vascular health, then we're going to have better brain health, and that's very diet related.
Thank you for that. Thank you for that question. Yes, there's somebody down here in the third row. In the third row, just here. Thanks. Near the aisle. Thank you.
Hi, I just wanted to make a bit of a comment or observation. I've come here to learn more about the brain as a retired medic. I have a son who has fetal alcohol spectrum disorder, and that's seen, to use your gardening analogy, as having lots of ponds in the garden, so you can't actually even plant roses. But it's quite clear, he's 25 now, that they do actually learn. But I wondered, if you knew people that were doing it. I think there's also a real association with autoimmune disease. He has very bad autoimmune thyroiditis. So I'm very interested in the second part, to come back to what, again, could be used to try and improve matters. But I just wonder about the effect of alcohol on the microglia, that because I didn't realise it could go right back in utero like that, so that even when they're starting off their brain cells and their brain formation, there's a fundamental not able to prune it properly at all.
Yeah, thanks for that. So
Yeah,
would that make sense?
Yeah, of course. So there is quite a few in people epidemiological evidence that in viral infection or any kind of really robust or too much of an immune activation can induce, in pregnant moms, can induce brain miswiring, even in utero, right? And so you're absolutely right. So that is a crucial part of research right now, and now using large-scale genetic studies where people try to find out what kind of genes which are, the DNA basically barcodes, right, to how our brain is shaped or our body is shaped. Now we're finding out that there's this immune genes that are implicated in increasing the risk for autism as well. But it is relatively new new research, right? Where we're beginning to pin out mechanistically how does the immune system impact brain wiring. And as I mentioned, complement is part of it, but it's just part of the story. We also know, as Lorena mentioned, cytokines also play a role in there. But yeah, so hopefully we'll figure out soon.
Thank you for that. Do we have another question before we continue? Yes, one in the second row just here. Oh, and apparently there's one at the back as well. Yeah. There's somebody just here. Can you wave to them so they see? Thanks.
Thank you. I have a question. Lorena mentioned, I think in one of the slides, MS, and I think we know that autoimmune diseases are more prevalent in women than in men. Is there any association also with neurodegenerative diseases that you have seen differences in the immune system or something?
Do you mean specifically with MS, or are you talking about with
any autoimmune disease?
You're talking about female bias?
Yes.
So there clearly is a female bias, at least for Alzheimer's disease, and I think some of the other dementias as well. And that, for a long time, we kind of dismissed it as just because women live longer. Us men drop dead in our 70s, unfortunately, and women tend to live 10 years or so longer. And therefore, the increased incidence of dementia was seen as they lived longer, and as aging is the biggest risk factor for Alzheimer's disease. That was the reason. What's now clear with very big population studies is that's one factor, but there's another factor, and women definitely have an increased risk of Alzheimer's disease, for example. And that is thought to be a hormonal factor, but we don't fully understand it.
So you don't think it's related to the immune system, and that's why they're
Oh, it might. The disease process will be immune related, but it's the changes which initiate that immune attack or dysregulation in the brain that we don't fully understand the reason why women are more susceptible to those than men.
Well, that takes us on rather nicely,
I
actually.
can add to that, sorry. So this is an active area of research of mine, and we are looking at the response of microglia to amyloid beta in Alzheimer's disease. And we do see that the female microglia and the male microglia respond completely differently. Well, completely is a strong word, but they do respond quite differently. The microglia are more inflammatory, and they respond in different ways to the amyloid beta protein. And this also happens in other diseases that are immune diseases, other macrophages in the lung. There's also been reports that female macrophages are more responsive. And just like Paul mentioned, this might be hormonally driven. We have evidence to suggest that it's actually chromosomally driven as well. So that's an active area of research of ours, and I think that it's going to be quite important in developing therapies, right? Because we can't develop a therapy that's going to fit all. If males and females are responding differently, we need to tailor that information for the development of therapies.
Yeah, Soyan.
Yeah, and we also know that male microglia, right, microglia from males versus females also mature differently during development. So people who are studying development have pinned out using, sequencing studies that their maturation state and the kind of genes and proteins that they make are different according to their age. But it's not all gloom for women. In Parkinson's, actually the reverse. Men are more susceptible.
That's so interesting, though, about the male and female glia, microglia. That's really interesting. Hold your other questions for the moment, because actually this takes us on rather nicely to what we want to talk about next, which is the types of diseases of the brain that can be triggered by the immune system. So Paul, which types of diseases overall might be triggered by the immune system, but that are then diseases of the brain?
Well, obviously, we've already just mentioned the classical autoimmune disease of the brain, multiple sclerosis, and there are variants of MS-like diseases. And there, what we clearly have is an autoimmune attack on the brain. So that's very, very clear. We understand it. In MS, it's an attack on those cells that make the insulation that Lorena pointed out to you, the oligodendrocytes. And loss of that insulation leads to the problems that people with MS have in terms of conductivity loss in their cells. So that's the obvious one, and there's a family of autoimmune diseases which fit that very well. And as I said earlier, even 15, 20 years ago, we didn't think of dementias, Alzheimer's and Parkinson's as being anything to do with inflammation and immunity. We thought of them as being degenerative diseases. That's hence we call them neurodegeneration. And now we know they're not degenerative diseases. The degeneration we see is the endpoint of the disease. The action is the inflammation, the immune damage that occurs in these diseases, probably decades before symptoms are even evident. And this is a problem we have with treating dementias like Alzheimer's, my own area of interest, is we need to be able to treat very early before the damage is done. So we need better tests, better ways of predicting the disease, and then treat people with the right sorts of drugs to dampen inflammation long before they start to develop dementia. Because probably by then, as they say, the horse is long bolted and there's no point in shutting the stable door.
So would that probably be long before people had symptoms at all?
That would be the ideal time to do it. At the moment, we're kind of in a position where we don't really have the tests, but the tests are improving absolutely phenomenally quickly. So we now can predict, or at least pick up very, very early pre-symptomatic dementia from a blood test. We couldn't do that even 5 years ago. We have better scans and so on to pick up early dementia. So we can pick up now people at the very earliest stages, and if and when we have effective drugs, treat them long before the damage becomes severe. We want to go back even further, treat in a preventive manner rather than a treatment way. That means getting tests which tell you who's going to get dementia before they get it.
Wow, yeah, amazing. And this makes me think, Soyoung, could neurodegenerative diseases in a way be classed as autoimmune diseases? You know?
So I don't know if Paul will agree with me, but I would say no, right?
Right.
The reason is, so what is clear, I get this question a lot, you know, is Alzheimer's, Parkinson's autoimmune? I don't think so, because I think for me, autoimmune is very clear. It's clearly the immune system being the triggers and the causes. But I think neurodegeneration is more complicated than that. I think it has to do with the analogy that Paul mentioned at the beginning of our discussion, the garbage, right? I think the garbage needs to be placed to activate or to trigger the immune system to clean up the garbage, but it's not doing it properly, right? So I hope that makes sense. So I don't think the immune system necessarily the cause of it, but definitely if we boost the immune system, we can slow down the degeneration process. Yeah.
And Jessica, what about the nervous system more generally, not just the brain? What kinds of infections might cause inflammation there, and what kinds of diseases would we be talking about there?
Yeah, the nervous system is everywhere in the whole body, so it's definitely not just in the brain. And in the body, there will be exposure to microbial infections, bacterial infections, viral infections. But as we talk about the brain today, you know, the blood-brain barrier is actually perfect system to keep all those infections out, and it does it very well. But bacteria and viruses do enter. They can cause meningitis, which is inflammation on the surface of the brain, which is a very dangerous condition to have. You can also have viruses entering the immune system, a macrophage, for example, and then hijack into the brain. So viruses can get into the brain through the immune system. And diseases such as COVID, the virus can enter the brain, but what it really does is to affect the blood vessels. And then the blood vessels become inflamed, and the inflammation associated with the attack can then influence the brain.
So it's not necessarily a local infection in the first place, it can be from somewhere else.
Yeah, and then there is inflammation in the periphery itself, which we know can trigger these signals to the brain that makes us feel ill. And if you already have an ongoing or early stage of dementia, then those immune triggers can accelerate microglial activation, and that's very damaging to the brain. But it's not caused directly by those bacteria and viruses, but obviously they're the trigger of those.
And Soyoung, we've been talking a lot about neurological conditions, but earlier Lorena mentioned mental health as well. Do we know how the immune system might be having an impact there, and how inflammation plays a part there?
Yes, I think it is similar to the response I gave to the question about autism. So we know from this large population studies looking at, you know, genes of people who are infected with schizophrenia, for example, that people are finding a lot of these risks, the risk genes, right? The genes, if you have mutations in these genes that induces increased risk for schizophrenia, for example, are also linked to the immune system. And going back to complement, which is, I think, a common theme that we're talking about today, complement actually also has been shown to, mutations in complement factors have been shown to increase risk for schizophrenia. And that's an active research of one of my mentors, actually. She's studying how, you know, dysregulation of complement leads to increased risk of this of schizophrenia. And it goes back to the role of microglia and synaptic pruning. So what we're thinking is that there is this, you know, either an overactivation or lack of proper activation. I think that's the most proper word, lack of proper activation of complement to regulate these, you know, microglia eating proper synapses, and that can lead to symptoms, at least in mice, are close to schizophrenic-like symptoms.
It's really interesting. So Lorena, do you think it does seem as though if it can play a part in mental health conditions as well, it's almost as if inflammation is everything, you know, that we really need to work out how it is to get this inflammation to be the right amount and not too much.
Yes, it's a very fine balance, right, between it being helpful or harmful. I don't think it's everything, obviously. We can't say that everything can be solved by solving inflammation, but I do think that it paints it's a very big part of the picture, and it's something that's been completely ignored until the last couple of decades. 20 years might seem like a lot, but in science, that's very, very recent. So yeah, I think it's not everything, but it's definitely something that we need to keep an eye out and investigate, especially in neurodegenerative diseases. They're coming up all the time. The risk for Alzheimer's disease is very heavily carried by microglia. So originally, what we knew about Alzheimer's disease and the genetic risk was based on familial forms of Alzheimer's disease, and that pointed at the neurons and this amyloid beta, but that only accounts for less than 5% of cases. Most of the cases of Alzheimer's disease are sporadic, and the risk there is linked to mainly microglia.
And we have, sorry, yeah.
I just want to add, but it's not only on microglia, right?
No, of
so
course
what
not.
What is fascinating is we're finding that these immune genes, because people, I think, neuroscientists, would traditionally focus, which I'm one of them, traditionally only focus on microglia, but now we're beginning to understand these genes that modifies the risk of someone developing Alzheimer's disease are actually expressed on T cells and B cells and NK cells and these other immune cells that we thought are not relevant to the brain. And so that is what's so fascinating in our field right now is that, because I do agree it centers on microglia function, but what is becoming increasingly clear that these peripheral or the circulating immune cells also impact microglia function as well. So we really need to look beyond the brain to understand brain disease.
And I think just to follow on from that, I think this link to T cells is quite an interesting one, but it's still true that they only infiltrate the brain very late on in the disease. So we do have the question of, is it the signaling from the periphery that is changing something in the brain, or is it something within the brain? And then later on,
I see, which comes first is what you
timing is everything, and
Yeah.
these diseases, as Paul mentioned, you have the symptoms much later on than when the disease actually starts. And so it's just a minefield out there. We need to understand the timeline of everything that's happening before we can understand the disease properly.
Yeah. And Jessica, you wanted to come in there.
Yeah, I would like to bring the gut in again.
Yeah.
Because the gut and diseases such as autism and also mood disorders, anxiety, depression, there's now increasing evidence that the type of bacteria in people that are affected by those conditions is different than from people who are normal healthy. And that's very interesting because these bacteria that are lacking perhaps are responsible for generating hormones and immune regulators that we need for our normal brain function. So when you ask, are mental conditions, mood, or this relate to the immune system, they're very much so, but it's maybe the bacteria in your gut that contribute to that. And there's one amino acid called tryptophan, which we have to get from our food. We can't make it ourselves, we have to get it from food. And bacteria in our colon, in the lower part of our gut, they digest fiber and then they generate these hormones. And immune cells make those as well, and they're directly interactive. So if you haven't got those microbes, you don't make those amino acids, and they affect your mood.
And one of the neurological diseases that we haven't talked about much so far is Parkinson's disease. And there's also research on the gut there, isn't there?
There's good evidence that the composition of the microbiota in Parkinson's is different. It's not as diverse as a normal healthy microbiota. And the bacteria that are lacking are those that make small fats. They're called short-chain fatty acids, and they have an anti-inflammatory effect. So as a consequence, by not having those bacteria, you're automatically more inflamed.
But do we know whether that comes first, or is that a consequence of having Parkinson's disease, those changes?
Well, thank you for the question, because there is some evidence that 20 years before you get the symptoms of Parkinson's, there are changes in your ability to have bowel movements, and constipation is one of the first symptoms that seems to be linked to developing Parkinson's later on. So that's really interesting evidence from humans. There's also evidence from people that inflammatory bowel disease, which is also associated with inflammation in the gut. And as a treatment, people get anti-TNF inhibitors, which inhibit inflammation. And there's some evidence, we need more evidence for that, but those that are on those anti-inflammatory treatments, they have a reduced risk of developing Parkinson's later on in life. We need more evidence, but that's again evidence that.
So in terms of implications for treatment or prevention, I know there is no such thing as a single ideal microbiome, but because we've all got different ones, and there could be many that are good, as well as ones that put you at more risk. But would there be a way of working out then perhaps how you could change everybody's microbiome, or the microbiome of those who you think are more at risk, in advance, way before, in order to try to prevent Parkinson's?
Well, it's a very interesting question, but hugely complex to answer. And it's not easy to change the microbiome, as Paul already mentioned. Just simply eating a yogurt is not really going to solve the problem. So we need to study it in a lot more detail. But because diet can influence the microbiome, it's one thing we can do to modify it. And it might be easier to do that compared to pharmacological therapies.
So thinking about treatments, Paul, I know you're working on something that's very cutting edge for Alzheimer's, these anti-complement drugs. Can you tell us about those? What does that mean?
Yeah, so anti-complement, we've talked about the complement system, and for me, complement is synonymous with inflammation. When complement's activated, it causes inflammation. It is all the garbage and all that, and bacteria, fine, but inflammation is what it does to cause pathology when it's overactivated. And we have lots of diseases, not in the brain, peripheral diseases, muscle diseases, diseases of the blood, where complement is known to be the driver. And what we have already in the clinic for more than 15 years now are drugs to inhibit complement in those systemic diseases. So kidney diseases, blood diseases, and a number of others. They're in the clinic, they've been transformational in those diseases. They have cured diseases which had no cure before. It's just an amazing change in 15 years. The problem is none of these drugs get into the brain, so we can't use them to treat brain diseases. So they're either big antibody molecules, which don't get into the brain, and Jessica and I both work on antibodies, or they're other small molecules, tablet-type drugs, but they, again, don't get into the brain. So our work has been really focused on getting the drugs, whether they be antibodies or small molecules, designed such that they are anti-inflammatory, but can also get readily into the brain. And so that's where we've been going. So these are drugs targeting the complement system because it's what I work on. But because they target complement, inhibit complement, they therefore dampen down inflammation very efficiently.
And where have you got to with those? What stage are they at?
We have, it's the usual thing, we're all scientists. I can tell you they work in mice, and they work very well in mice. We are at the point now where we are spinning out a company in order to, it's impossible for an academic lab to take a drug to the clinic. It just, the money isn't there, you can't do it in the way that we do our research. So what you've got to do is get into the point where people are interested enough to fund a company, spin out a company, then the drugs get made, and eventually they get to the clinic. So maybe in 10, 15 years, because that's what it takes, these drugs will be in the clinic.
Yeah. Yeah. Now, there is, of course, a class of drugs for Alzheimer's disease, which have been, in the news a lot, drugs with names like lecanemab and donanemab. Jessica, can you explain how those work? What are they doing? Where are we at with those?
Yeah, so those are new biologic drugs. They're antibodies, and they bind to the amyloid beta plaques that Lorena introduced in her presentation. So the amyloid is forming plaques, and they cause some of the changes. And so there's a hypothesis called the amyloid hypothesis that states that amyloid is the starting point of all the downstream problems. So having an antibody which would allow binding and then recognition by the immune system is a very efficient way of removing the amyloid from the brain. And it works really well. It works in mice and in humans, so it's a bit further developed. The problem is that it doesn't work very well, and there are various different reasons for that. So there's around 27% delay in the progression of clinical symptoms when you have those class of drugs. So there's a lot of people who don't respond to that. And there's various reasons why that is. Now, I think we should really say that it is the first time ever that we can change the disease with drugs. There is no other drugs for Alzheimer's that can do that. So I think the message here is that it's a fantastic discovery that we can do this, but we have to do better.
And is it stopping future damage?
Yeah, so the antibodies,
slowing the future damage.
They do. We probably need longer time to analyze those studies, but there is now evidence that they delay the progression. But they don't get very well in the brain, so that's one problem. When they do get in the brain, they may cause some inflammatory side effects. So that's a problem as well, because you have to lower the dose, and it's a problem. It requires monitoring. Every person who gets the drug needs monitoring. Brain imaging is not readily available in England or in Europe. And so that is a problem for licensing these drugs. But we do have technologies. We can change antibodies. We can engineer them. So this is only the beginning. And because we've now got evidence that you can modify the disease, it's only a matter of time where those type of drugs will be effective.
Yeah. I mean, Lorena, I'd be interested to know, because you're working in this area of amyloid and so on, what potential do you think these drugs have, or the future of drugs that might come?
So I think they have huge potential. I think everything, this is doing it again. Everything that Jessica said is correct. These antibodies find it difficult to get to the right site, but actually we can engineer them and make them better. So Roche, for example, is a pharmaceutical company that's now trialing a new form of these antibodies, and they've modified them so that they can more readily penetrate that blood-brain barrier and get to the brain. And apparently they can reduce the levels of amyloid in 6 months. So they're much quicker, the new generation. So these two are the first ones that have been released, but it's also starting a whole new area of investigation. I think timing is very important, as I already said. So these antibodies, they're being, the clinical trials were given to patients that had early Alzheimer's, but that's measured by the dementia. And as I said, the amyloid buildup has been happening for many years. So the future, what we're trying to think about is that, as Paul mentioned, you can now have these blood tests that can detect whether you're going to get dementia much earlier on. And ideally, you would be trying to give those drugs very early on. So as soon as you have a positive blood test, you give these drugs, and maybe then we can have much better outcomes than we have at the moment.
I guess then that would be difficult if there's difficult side effects with them and risks.
Yeah, but the side effects, I think, they're less than 1% of the patients that are getting the side effects. And I think, again, it might just be to do with the timing of it. And I think that the drugs are improving, right? So, of course, we have side effects, and they are very scary drugs, but the basis is there, and now we can build up on that, right? We can try and design better drugs based on what we know.
And before we open things up for questions again, there's one other thing I want to ask you, Paul. What about, because of the involvement of the immune system, does that imply that you could have some sort of vaccine that could be developed to protect against all sorts of different neurodegenerative diseases?
It would be nice to think so. I don't really think it's a realistic prospect in the near future. I should say, long before the anti-amyloid antibody drugs came along, there were studies of immunizing to remove amyloid. So basically giving people injections of an amyloid-like protein to get them to make an immune response to it, like any other vaccine, in order then for the amyloid to be cleared from their brains. And these were actually very good at clearing amyloid, not quite as good as the antibodies that we now have, but they were very good. The problem is they caused huge side effects. They caused neuroinflammation, and people who were given these, and these went into clinical trials, the immunizations, were dying. And of course, the problem when you've immunized somebody is not like a drug. You can't just stop it. You've done it, and you've got to wait for the consequences to pay out. So I think that's scared people a lot. I don't really envisage a simple way to clear amyloid. Of course, we've got some interesting things around infections and dementias, and we now have this really interesting story coming out from the shingles vaccine, which I'm sure many of you will be aware of, where getting the shingles vaccine in your late 60s or 70s gives you a really big reduction in risk of getting dementia in the next decade. Something
That's
like
really
20 to 30%. It's fascinating.
Yeah.
And again, we don't fully understand. There's 2 or 3 hypotheses out there, but likely, I think, this is about clearing the herpes virus from the brain and removing the virus, which may be causing some chronic inflammation. And so getting less chronic inflammation, less risk of dementia. So there are ways we can do these things, but it's usually going to be targeting a very specific factor in the disease.
Really interesting. Jessica, you nodded there, yeah, and then we'll go to questions.
So we talked a lot about therapies, and it's a hugely exciting time to think about therapies with some successes as well. But what we should also probably think about is prevention. Prevention is better than a cure. And over the past 10, 15 years, there have been a number of risk factors identified that are modifiable. And actually 45% of all cases of dementia can be explained by a modifiable risk factor. And with that, I mean your diet, the level of exercise, where you live, various different risk factors. 45% is significant. And so if we can take these into account, along with therapies, perhaps a combination of those is the future way.
Yeah. Thank you for that. Let's take some questions from people watching at home. Yes.
We've had lots of questions from
Yeah.
online. There's a great one from Francis, which I don't think we've covered. So if the brain is making waste, how is this normally removed from the brain?
Yeah, good question.
How is it normally removed? And if it isn't cleared,
Right.
does this affect dementia and inflammation?
So the brain does make waste, and we during aging, accumulation of waste in the brain is seen normally. Every brain one looks at pathologically from a person of my sort of age will be full of little aggregates of garbage called corpora amylaceae. These aren't amyloid plaques, they're different, but they're the normal accumulations of garbage in the brain. Most garbage is cleared, and it's cleared by complement microglia doing their job of taking out the trash, and the microglia just basically eat what they take up. They phagocytose and consume that, but you will still get accumulation of garbage. In pathology, though, the buildup of these abnormal waste products like the amyloid plaques and tau tangles and so on is much harder to clear, and therefore it will accumulate, and the microglia cannot deal with it just on their own.
Thank you. Do you have another question from at home?
Well, I think Soyeon's going to expand.
Yeah, so it also happens during when we sleep.
Oh, yeah.
So every time we sleep, our brain gets rid of the waste, right? So this is why what our parents say is all true. Sleep. Make sure you sleep your full 8 hours.
Yes. Do you have another question from online?
So this is a bit different, but someone has asked, after COVID and all the disruption that COVID has caused to the immune system, do we think there's going to be a lot more cases of dementia or brain diseases going forward?
Interesting.
I work on long COVID, so perhaps I should take that. And it's a scary scenario, it really is. And we have a huge number of people who have long COVID. One of their major symptoms in most of them is brain fog, and this kind of fugue state that many of them get. And this is exactly the sorts of things that we know can increase the risk of dementia. So I think we will probably see a tick up in dementia as these people age to that time period in their lives.
Who else has a question from within the room? Do wave. Yes, one right down at the front here. Oh, and you've got the mic to one there. Fair enough. We'll go there first, and then we'll come to you.
Hi, hello. So I've got fibromyalgia and family predisposition to dementia and bowel conditions, and I'm awaiting investigation for ADHD and autism. So my interest is, like, how do these things come together? How do you assign weight to the different aspects? So even like with improving diet, exercise, and psychological triggers, like it's very much still present. So, but which treatments are working? How do you, where's like genetics start and continuous factors in like behavior and environment? Like, how do you weight that, and how? So
Yes, it's a huge question, really, isn't it, about what the causes are of everything, which, of course, are interconnected.
So many things contribute to risk. I mean, genetics is a huge thing, and we, from 20 years ago, again, having one genetic risk factor, we now know at least 100 genetic risk factors for Alzheimer's disease, and you roll the dice and you get the genes that you get. So that's something you can't really control. Things like chronic inflammation, you say fibromyalgia. Well, yes, of course, there is an association, but actually, if you're on anti-inflammatories, long-term anti-inflammatories, you have fibromyalgia, that reduces the risk of dementia. This has been shown in lots of prospective studies. So just taking non-steroidal anti-inflammatories has an impact. And of course, aspirin is one of the best things to take in low dose, because that's again been shown to improve vascular health and lower dementia risk. So there's a lot going on, and you just got to accept that some things that happen to you may increase your risk of dementia a bit. I played rugby. Gosh, I don't know what that did to my brain. So we have these things that we have to live with, and it'll have a little effect, but the main thing is stay healthy, use your brain, and you hopefully won't lose it that way.
Thank you. And there's somebody with a question in the front row, just here, right at the front. Here comes the microphone, just here.
Can I ask a question on lupus to Paul? Can I ask a question on lupus? Yeah. I know a young person in her late 20s who's been diagnosed with lupus. What best thing can she do? In terms of diet, in terms of treatment, in terms of...
I mean, lupus is real lupus, systemic lupus erythematosus, SLE, the chronic lupus, is an autoimmune disease. It's one of the more difficult ones to treat, and it does respond to the usual things we treat autoimmune diseases like steroids with. There are new therapies coming along, biologicals, some of which seem to be showing a good effect in lupus. So I think there will be new and better biological drugs for lupus in the future. The sorts of drugs that have already come to the market in other autoimmune diseases like MS, for instance. So I think the scene will improve. It's a difficult one to treat, but it is an autoimmune disease, and we know how to treat those.
You talk about steroids, but steroids damage the immune system.
Absolutely do. And steroids are wonderful drugs, apart from what they do to you. They cure the disease, but they can make you very sick afterwards. So what we need is drugs that are as effective as steroids, but don't have all the side effects. And a lot of the things we take into autoimmune diseases now are what we call steroid sparing. So you still use a bit of steroids, enough to have an impact, but you also have another drug which lowers the amount you have to use, so less side effects.
Thank you.
Now,
Thank you very much
made a reference to yogurt, but she's taking like yogurt with reuteri. You know that? You know this kind of things?
I'm not a fan. I do have yogurt for my breakfast, but that's because I like it. I don't think it's going to make a huge difference to my immune health. And I think there's a lot of hype on these things, and often very expensive things. So I'm not convinced.
Thank you for that. Who else has a question? Yes, somebody in the middle just here. Oh, you've got somebody with the mic already, ahead of me. Thank you.
My question is around the gut and brain relationship you've talked about. In terms of, I think, Jessica, you mentioned that it's controlled by feelings. So in terms of the feelings, if our thoughts are impacting the gut, is there a way that it could be improved or addressed by our thoughts or change in feelings? And the other thing is, how do you stop a leaky gut?
What an interesting question. Thank you. There is evidence that the gut talks to the brain and the brain talks to the gut. But the gut talks more to the brain than the other way around. So the changes that cause these feelings, changes in feelings, are driven by a nerve called the vagus nerve, which innervates all our organs, but particularly the gut. And changes to the triggering of that nerves can be induced by changes in bacteria and the release of chemicals that those bacteria produce, or the lack thereof. Immune cells also release chemicals and cytokines, and they communicate with the brain through these neural systems. And if you don't have enough of the bacteria that produce those beneficial chemicals, then the brain will be affected. And there's evidence that that changes your mood. The tryptophan, which are in the serotonins, if you don't have enough of those, then they will affect how your brain works. How do you prevent a leaky gut? You can prevent a leaky gut by a healthy diet. If you eat fibers or fermented foods, the bacteria in your gut, they really love those. We can't digest those ourselves. We need the bacteria to do that. And they produce short-chain fatty acids, which are small chemicals that promote a mucus layer in our gut, and they prevent the gut from becoming leaky. So eat a healthy diet.
Thank you for that. I want to fit in a couple more questions because we're very much running out of time. Yes, there's somebody at the back, in the middle up there, near the back. Thank you. Yes, we'll go to there and then we'll go to the back.
Sorry, I'm not at the back, I'm the middle.
That's all right. We'll go to the back after.
I have a question. Do any of your research cover transcranial pulse shockwave in relation to Alzheimer's and how the inflammatory process can be reversed with the anti-inflammatory first, or whatever it works?
Anybody know this?
I've not experienced using it or working with people myself, but we do have some people in our institute who use these transcranial stimulation type things in tandem with imaging. So they're actually focusing the stimulations to very specific areas of the brain. And in those contexts, and particularly in Parkinson's disease, this can be very effective. At least for short term. I think the long-term results we're still waiting for, but they certainly seem to give a bit of a kick to areas of the brain which aren't working very well.
Thank you for that. And then there was somebody in the middle at the back who I promised to go to up there, right at the back. Could you wave so they can see you? Thank you. Thanks.
Hi, you mentioned there's now a blood test to test for Alzheimer's. I was just really interested, I wasn't aware of this. What is that blood test actually looking at to indicate that Alzheimer's is on the cards?
Sorry, I'm not really trying to dominate, but biomarkers is my thing. We've been very fortunate in the UK with the DRI. We have a biomarker lab at UCL, which is world leading, and they have really managed to set up great tests from blood samples for predicting particularly Alzheimer's disease. What they're looking for fragments of amyloid, the protein that accumulates, and they're looking for very specific fragments of tau, the other protein that accumulates. And a combination of amyloid markers and tau markers, and a couple of other things like markers of neuronal injury called neurofilament light chains, can give you together, when you put them all in a package, a very good prediction of risk of getting dementia, even pre-symptomatic. So at the moment, the tests are still quite expensive because they require very high sensitivity analyzers and so are limited to a few places. But I'm sure that's going to change and we'll have these sorts of blood tests available much more widely in the next few years.
I'm very struck by how each of you have said how much has happened within the last 20 years. And I'd like to ask each of you, what do you think will happen in the next 20 years? You know, how fast is this moving? Are we going to get some real treatments for all of these different conditions that we've heard about that are going to be useful, or some of these different conditions? Lorena?
Well, I think that the field is, as I said, exploding, and I think there's a lot of hope for the future in treating these diseases. I think in terms of therapeutics, 20 years might still be too short. As Paul said, it takes more than 10, 15 years just to get something to the clinic. So it's going to take a bit longer than that. But we're learning something new every day. These blood biomarkers that we've just spoken about, they only came out a year ago. They were published a year ago. Immediately, there's been this big clinical trial that's taking place at the moment. So the field is moving very fast. So I think it'll be hopeful for the future.
Soyan?
Yeah, so for me, I think it's from the scientific front, I think, you know, neuroscientists have to work more closely with traditional immunologists to really understand how the immune system shapes the brain during development as well as during early stages of neurodegeneration. I think what will truly change the field is when we develop biomarkers, peripheral biomarkers that we can detect way early, you know, before we start seeing memory disorders in patients.
So ways of being able to measure that people have got
this. And I think the immune factors that we can detect in our blood are going to be it. But the key thing is specificity. And so this is where I think scientists from different fields need to come together and do it. And so I think in about 10 years from now, that's where we'll be, you know, changing the immune system to change the brain, because it's not only a brain disease. That's what we learned here.
And Paul?
Yeah, I think the comparison of where we are, where we've gone over the last decade with Alzheimer's compared to where we are with cancer is really sobering. There's been a huge investment in cancer, enormous amounts of money have been spent, and we are seeing the drugs, and particularly immunotherapies now for cancers, changing the outcomes hugely for people with cancer. So we need to catch up in dementias, and we need to be doing those sorts of big investments to get these drugs, immunotherapies and others into dementias, because these are the two big health challenges for the future, and we're hardly scratching the surface yet for dementia.
And Jessica, what would you say?
I'm also very hopeful for the therapeutics, but I'm also hopeful about the prevention, because we are starting to learn much more about diseases being a whole system condition. And the gut immune brain axis, again, I think is a huge opportunity to start learning more about these conditions. And I'm very proud that we're launching a UK-wide network where academics, policymakers, innovators, the food industry, people from the public can all join up in a network and talk to each other and do the things that Soyeon says, like immunologists talking to microbiologists, microbiologists to neuroscientists. Let's do this together, and I'm hopeful that will happen.
Well, I think you've given us food for thought there, and some optimism as well, and it's so interesting to see what's happening. I can't wait to know in 20 years' time what is going on. Thank you so much to our panel, Jessica, Paul, Soyeon and Lorena, and also to the many people who've organised this event, Anne O'Gara, Andreas Wack, Katano Rice-Souza from Crick Research, Rosie Waldron from Crick Public Engagement, Louise Howard and her team here at Crick Events, Yvonne Borden and Ursula Weiss from Nature, and Jenny Evans and Doug Brown from the British Society of Immunology. And thanks to those organisations for funding this tonight. But thank you so much for all your very intense listening there. I'm Claudia Hammond. Goodbye. Thank you.